Okay guys, now after discussing about lipids, now let's discuss about proteins. Okay, we have seen that cell membranes are made up of lipids as well as proteins and carbohydrates. We have seen different types of lipids, phospholipids, glycolipids as well as sterols. Now we are discussing about the proteins. The most important point about the proteins is proteins are major components. Okay, they are the major components of the cell membrane, and they are accounting for, why they are calling as major component, because they account for 55% of the cell membrane. Now guys, please concentrate here. You can see different types of proteins, right? In the bilipid layer, you can see here there is one type of protein, here there is a different type of protein, there is a different type of protein over here, and you can see one more type of protein. So there are different types of proteins which are present in the cell membrane. You can see here GPI anchored proteins. You can also see peripheral proteins. You can see integral membrane proteins. So what I want you to know is there are different types of proteins. But right now let's discuss about the most important types. Okay, types of proteins. The first type include transmembrane protein. Okay, transmembrane protein. The second type include peripheral protein. And the third type include lipid anchored proteins. Lipid anchored proteins, three different types of proteins. Now, what does it mean by transmembrane proteins? Transmembrane protein or integral proteins are the ones which are passing throughout the length of the cell membrane. Now, throughout the thickness of the cell membrane, these proteins are passing from outside to inside. So, these are transmembrane, they are passing throughout the membrane. Okay, so they are extending from outside to inside. So, they are called as the transmembrane proteins. Okay, if I have to show you cell membrane something like this. Now these proteins are extending from outside to the inside. Here is the cytoplasm. Here is the outside environment of the cell. So from outside to inside, these proteins are called as the transmembrane proteins. Now peripheral proteins. Peripheral proteins are only present in the peripheries, either on the outside or on the inside. Peripheral proteins, either on the out or on the inside, towards the outside of the cell or towards the cytoplasm. So these are the peripheral proteins. And third type of proteins are the lipid anchored proteins. Now these proteins are attached to the cell membrane with the help of a lipid. Okay, they are attached. You can clearly see here. See, GPI anchored protein here. Okay, this is an example of lipid anchored protein, a protein which is anchored with the help of a lipid. Okay, now we have seen three different types of proteins. Now we have to know what are the examples of these proteins. Okay, let's write down here. The examples of transmembrane proteins are receptors. Okay, we all know receptors are there, right? Now receptors for the hormones. Now these receptors are examples of transmembrane proteins. Second examples are pumps. Okay, pumps means these are the ones which are allowing the movement of ions with the help of ATP. By breaking down ATP, they are allowing the moving of substances. Okay, pumps. Example, if you ask me example, the example is sodium, potassium, ATPase. So sodium, potassium, ATPase is an example of a pump. Now pump is a which type of protein? It's a transmembrane protein. So receptors, pumps. The third type of proteins are ion channels. So transmembrane proteins include receptor, pumps and ion channels. Okay. Now, what are the examples of peripheral proteins? Peripheral protein examples are spectrin, ankyrin and dystrophin. Okay, and lipid anchored proteins examples are GPI. Okay, GPI is an example of lipid anchored protein. Now this GPI molecule is present on the surfaces of cells. Now it's having, imagine it's a protein which is having two hands. With one hand, it is catching a molecule called as DAF, decay accelerating factor, and MIRL, MIRL, membrane induced reactive lysis. Now what is the importance of this? See, this GPI is an example of lipid anchored protein. Now this lipid anchored protein, now it's having two hands. Imagine it's having two hands. With one hand, it is holding DAF on the cell as well as MIRL on the cell. So on a cell, if I have to show you, imagine if this is a cell. Now on the cell there is this one protein called as GPI. Now this GPI protein, let's write it down here, this is GPI. Now this GPI protein is having two hands. Now with one hand it is holding DAF, decay accelerating factor, and MIRL, MIRL, membrane induced reactive lysis. The DAF, it's having a CD name, CD cluster designation of CD55. Okay, CD55 is DAF and MIRL is CD59. These are the cluster designations. Now what is the importance of this? We will see in a moment. Now let's discuss about some important points which you need to know for your exams. See, after seeing different types of proteins, now let's talk about spectrin. Okay. Now what is this spectrin? Spectrin and ankyrin are examples of peripheral proteins. Now where they are present? They are present in the cell membranes of RBC. Now what is the importance of this two proteins in the RBC? Now because of the spectrin and ankyrin, RBC is having biconcave shape. Okay, the biconcave shape of the RBC is because of the spectrin and ankyrin. So whenever you have defect in spectrin or ankyrin, that will cause change in the shape of RBCs. Now take down here guys, whenever there is spectrin defect, mutation, that will cause hereditary elliptocytosis, which means now the RBCs are not in biconcave shape. Now the RBCs become elliptical. Okay, something like this. Now whenever there is a defect in ankyrin, okay, ankyrin mutation will cause hereditary spherocytosis. Spherocytosis means sites means cells which are in spear shape. Okay, spherical. Okay, so spherical RBCs are the round shape RBCs. Now whenever there is mutation in ankyrin, the RBCs will become more of round in shape. So these are some important MCQs which you need to know. Spectrin defect will cause hereditary elliptocytosis. Ankyrin mutations will cause hereditary spherocytosis. Now after this, let's discuss about, we have discussed about spectrin, ankyrin. Now the last point which I want you to know here is dystrophin mutation. Why? Because dystrophin is also a peripheral protein. Now dystrophin is a very important protein which is maintained in the skeletal muscle structure. Okay, it's one of the biggest protein. Now dystrophin mutation will cause a disease called as Duchenne's muscular dystrophy. Okay, so Duchenne's muscular dystrophy is due to mutation of which protein? Peripheral protein. Which peripheral protein? It is dystrophin. Okay, different types of proteins and different types of disorders. Now, guys, I want you to know a very important pathological link here, that is paroxysmal, these proteins and pathological link. That the condition is called as paroxysmal nocturnal hematuria. The name itself is there, paroxysmally, abnormally, or sometimes there is nocturnal, during nights there is hematuria, blood in the urine. Why this is happening and what is the link, pathological link with the proteins which we are discussing right now. See, in paroxysmal nocturnal hematuria, there is a defect in GPI. These GPIs, which are lipid anchored proteins, which we have just discussed, there is defect in GPI. Now, whenever there is no GPI, there is no DAF and there is no mirror on the surface of the cell. Now, which cell I am talking about? I am talking about RBC. Normally on the surface of RBC, see, I am showing you here the healthy RBC. Now, this healthy RBC on its surface, it is having this pink color antibody shaped structure. Okay, it's not an antibody, it's actually GPI. Okay, this is GPI. Now, this GPI, it's holding two hands. It's having two hands. With one hand, it's having DAF and with other hand, it's having mirror. Now, what is the importance of this DAF and mirror? Now, importance of DAF and mirror is that now these two substances, they will inhibit or inactivate, okay, complements. Okay, they will inhibit the complements. First of all, now you will get a doubt. Sir, what are these complements? And now why this DAF and mirror are inhibiting the complements? See, understand, the complements are nothing but the proteins which are present in the blood of the humans, normally in you and me. In our blood plasma, there is something called as complement proteins. Now, these complement proteins are a part of immune system. Now, these are very good proteins which will kill the microorganisms during infections. So, complements, take it down here guys. Complement proteins kills bacteria, okay, or microorganisms. By formation, this whenever you activate the complement proteins by direct pathway, indirect pathway, which we will study in second year, in your second year pathology, whenever this complement proteins in your blood, whenever they are activated, now they will form something called as a membrane attack complex. Now, this membrane attack complex is the one which causes pores in the cell membranes of the bacteria. Okay. Now, imagine if this is a bacteria. Now, this membrane attack complex, it is going to drill holes in the cell membranes of bacteria. Whenever the cell membranes of bacteria, whenever they are becoming porous, now these bacteria will definitely die. But the important point is, now this membrane attack complex, this complement protein, now it's like a blind soldier. Okay, soldier is a good person. Definitely he is trying to protect the country. But this soldier, this membrane attack complex, it's like a bad soldier with a AK-47. Now, whenever he is activated, whenever you activate this person, he is going to fire. But whenever he is firing, not only bacteria will die, normal cells which are surrounding the complements, they will also die. See, these complements, whatever I am talking, they are present in the blood, right? Yes. Now, in the blood, there is RBC, there is WBC, there is a platelet, and there is also this, right now, there is bacteria. Whenever complements are activated, membrane attack complex is going to form. Now, this membrane attack complex, it doesn't know whom to attack and whom not to attack. Membrane attack complex should have to only attack the microorganisms, but it will also go and act on the RBCs. Now tell me, RBCs, whenever membrane attack complex is coming over them, what this RBC should have to do? Now these RBCs, healthy RBCs are having DAF and mirror. What is the function of DAF and mirror? Whenever this membrane attack complex, whenever an activated complement is coming towards them, now this activated complement will be inactivated with the help of DAF and mirror. I am just telling you in a very simple language. Okay, so the DAF and mirror which is present on the surface of RBC, WBC, platelets and all the blood cells, now they will inhibit the activated complements. Okay, but on the surface of microorganism, there is no such thing as DAF and mirror. There is no such thing as GPI. So now activated complements like membrane attack complex will go and kill only bacteria, but not the RBC, WBC platelets, because RBC, WBC platelets, now they are having this DAF and mirror. Now let's talk about a condition called as paroxysmal nocturnal hematuria. Now in this condition, paroxysmal nocturnal hematuria, what is the problem? The problem is defect in GPI. Now whenever there is this defect in GPI anchored proteins, there is no DAF is not there, decay accelerating factor is not there on the surface of RBC, and membrane induced reactive lysis, CD59 is also not there on the RBC. Whenever these things are not there, now they can't inactivate the complements. Now complements are not getting inactivated. Now you will get it out. Sir, first of all, why the complements are getting activated? See, usually during nights. What type of breathing you will be having? You guys or any person during nights will be having shallow breathing or deep breathing. The person like quiet breathing, right? Now he will be having shallow breathing, superficial breathing. Superficial breathing. Okay. Now there is no deep breathing, just shallow breathing is there. Now whenever there is shallow breathing, now the person is totally taking out the carbon dioxide from his lungs. Is he able to properly take out carbon dioxide from his lungs? No, this is just shallow breathing, right? There is carbon dioxide retention in the body. Okay, now during nights when the person is sleeping, yes, carbon dioxide is not properly getting washed out. Carbon dioxide is getting retained in the lungs. Now this carbon dioxide will react with the water, forms carbonic acid. Okay, carbonic acid. Now carbonic acid, right? Acid. Now during sleeping, yes, there is acidotic states in the body. Usually during night, there is acidosis in the body, and this acidosis can also activate the complement. True, today you go to sleep. Now during this night, the complements in your body are activated even without the bacteria, even without the microorganisms. Complements are getting activated because acidosis can activate the complements. Okay, so normally complements are activated during night, but in your body, when complements are activated, there is no problem. Why? Because your RBC, WBC and platelets, now they are planted with the GPI. They are having DAF and MIRL. So there is no hemolysis. But imagine if you are a person who is having paroxysmal nocturnal hemoglobinuria, your RBCs are not having, or your RBCs are lacking GPI. So there is no DAF, there is no MIRL, and the activated complements during night will come to the RBC, will attack the RBC, will cause mass destruction of the RBCs, that will cause hemoglobin to leak out of the RBCs, and that hemoglobin is coming out in the urine. Okay, whenever the person with a paroxysmal nocturnal hemoglobinuria, whenever he goes to urine in night, because of the hemolysis that is happening particularly at night, will cause hemoglobinuria. So that's the reason why we are calling this as paroxysmally nocturnal. During night, the person is having hemoglobinuria, red color urine, because hemoglobin is leaking in the urine because of the hemolysis which is happening during night because of the acidotic state in the body and due to lack of the GPI anchored proteins. So these are the important pathological links which you should know for your exams. Now after learning about these proteins, let's talk about the carbohydrates, guys. Okay, let's talk about the carbohydrates. See, carbohydrates are accounting for how much percent? Carbohydrates are accounting only for 5% in the cell membrane. Now, there are different types of carbohydrates which you should need to know for your exam, because see, these carbohydrates are used for blood grouping. Okay, especially these carbohydrates, based on the presence of different types of sugars on the RBC cell membrane, we have categorized the blood groups into A type, B type, AB type, O type, okay, something like that. Now, for example, if I am saying A type, what does exactly this A type mean? A type means this person's RBCs, on the surface of the RBC of this person, A group blood person, he is having a carbohydrate called as N-acetyl glucosamine. Okay, this N-acetyl glucosamine, which is also called as NAG, Okay, whenever you have this N-acetylglucosamine, then we can say this person is having A blood group. And whenever the person is having galactose sugar, okay, when the person is having galactose sugar on the RBC surface, now it's a sugar, right? Sugar means carbohydrate. Whenever the galactose sugar is there, then we can say the person is having B blood group. Then what is AB blood grouping? So AB blood group person have both NAG as well as galactose sugar. And who is O person? O blood group. O blood group means now this person doesn't have any either NAG or galactose sugar. Okay, this person is not having N-acetylglucosamine or galactose sugar on the surface of his RBC. So based on the presence of different types of carbohydrates, we have classified the blood groups into different types. Okay guys, we have discussed all the important points which you need to know for your exams regarding this one topic, cell membrane. Hope the video is helpful. See you in the next video with the new topic.